Good afternoon, everyone, and welcome to the first event in the new Women in Science Entrepreneurship Fund speaker series entitled Conversations with Women Leaders in Biosciences. As many of you know, my name is Elaine Fuchs. I'm the Rebecca C. Lancefield Professor and the head of the Chemers Neustein Laboratory of Mammalian Cell Biology and Development at Rockefeller. I'll be joined momentarily by Dr. Karen Akinsanya, who's kindly accepted our invitation to be our first guest in this series. Before introducing her, however, I'd like to tell you a little bit more about the Women in Science Entrepreneurship Fund, or the WEF as it's called, which is just launched at Rockefeller. So if we start by going back all the way to the Solvay Conference in Physics in 1927, Marie Curie was the only woman participant, shown here at the third bottom from the left. She deserved the invitation because at the time she had 2 Nobel Prizes. There are now 3 other female scientists awarded a Nobel Prize in physics after all of these now 100 years. 2 of them only in the last 2 years. So just a few years ago, the New York Times showcased an article on its front page reporting that when asked to draw an effective leader, most people drew a man. In biomedical sciences, changes come more rapidly. Since I began as an assistant professor, now some 4 decades ago, I've had the pleasure to mentor many women scientists who now run their own laboratories. Additionally, Rockefeller now has 13 women HOLs, comprising nearly 20% of our faculty. But there are still hurdles. So what's the next big one? The next big one is involving women scientists in translational science, biotechnology, and forming companies. So at Rockefeller, we've launched 8 different companies, and all of them are run by male HOLs. Many of the recent initiatives that are currently in the pipeline have come from the Robertson Fund. And the Robertson Fund has provided opportunities for faculty, students, or postdocs who first have a strong preliminary evidence of clinical or pharmaceutical application, and secondly, have the prowess of how to see it through. And this has really increased the pipeline for Rockefeller HOLs to establish more companies. But the problem is that those in the works are basically, again, HOL male dominated. And so how do we bridge the gender gap? Through the Women's Entrepreneurship Fund. And here, the WEF will provide competitive innovation grants to Rockefeller's women Ph.D. students, postdocs, and HOLs who have a great translational idea based on their early stage discoveries, and they're able to successfully defend it before a panel of biotech, pharma, and venture capitalist experts. So in order to achieve that, the WEF will provide entrepreneurial training to women at Rockefeller. In order to be able to do this, they'll also equip a new generation of female scientists then with the knowledge and skills needed to develop breakthrough therapies and diagnostics, working with biotech, pharma, and venture capital communities. And so I'd like to start by just mentioning a big thank you to the Women in Science organization at Rockefeller University and the early developers and supporters of the WEF, Rebecca John, Joel Caden, Denise and Michael Kellen Foundation, Marnie Pillsbury, and Robin Chemers Neustein and Lulu Wang, many of whom are in the audience today. One important aspect of this then is to announce the winners of the inaugural WEF grants, which were just successfully defended not only less than a week ago, And these are Agata Smorgazuska, Associate Professor, studying PIF1 as a therapeutic target in medulloblastoma, Sherry Cohen, a postdoctoral fellow in my laboratory, studying targeting lymphatic drainage stem cell-derived factors as a therapeutic target for lymphedema and enhanced wound repair, and Rebecca Timpson, a graduate student in Kavanch Bershoy's lab, studying the development of novel glutathione synthesis inhibitors. And this then brings us to the last and important aspect of the Women in Science Fund efforts, and that is to create this series of conversations that we can do that really bring in the best and the brightest who are out there and have managed to bridge that gender gap in terms of pharma and biotech. And so at this point, I'm really pleased to introduce Dr. Karen Akinsanya, and she's going to be our first woman WEF speaker. And so maybe we could shine the light on Karen for a moment as I introduce her. Hi, Karen. Welcome here.
Hi, Elaine.
So Dr. Akinsanya has more than 25 years of experience in a variety of fields, from bench science to senior management in the pharma and biotech industries. Today, Karen is the Executive Vice President, Chief Biomedical Scientist, and Head of Discovery, R&D, at Schrödinger, a company that develops state-of-the-art chemical simulation software for use in pharmaceutical, biotechnology, and other areas of research. Born and raised in the UK, Dr. Akinsanya received her Ph.D. in endocrine physiology from the Royal Postgraduate Medical School at Imperial College London. Following postdoctoral research at Imperial and the Ludwig Cancer Institute, Dr. Akinsanya moved from academia to pharma. Her first stop was Ferring Pharmaceuticals, where she worked both in the United States and the UK, and she led the discovery of a preclinical characterization of a chemotherapeutic agent, Firmagon, an FDA-approved treatment for prostate cancer. Following 8 years at Ferring, in 2005, Dr. Akinsanya took a position in the US with Merck Research Laboratories, and at Merck, she worked at a variety of different capacities, from directing preclinical and clinical trials to business development. Dr. Akinsanya joined the team at Schrödinger in 2018, and though Schrödinger is known for creating software and other tech solutions for drug discovery, the company has recently established their own drug discovery program, an area that Dr. Akinsanya now heads. In addition to this extraordinary career, Dr. Akinsanya has also made time to share her passion for science and technology with the younger generation. In 2010, she founded MyTech Learning, an organization based in New Jersey that provides grade school and junior high school students with the skills and confidence to excel in areas of STEM. So welcome, Karen. Thank you for joining us today. I'll be the moderator for a discussion with Karen, and then following this, there'll be a Q&A session with our audience. So if you'd like to ask a question, please do so. Click on the Q&A and type in your question at any time, and I'll try to get to as many questions as I can at the end of our discussion. So I have the pleasure to begin by posing a question for you. So many, I'm sure, in our audience today are graduate students and postdocs in the early stages of their career. And they're going to want to know, I think, going way back, what sparked your interest in science, maybe in your childhood upbringing, your teachers, who planted those early seeds?
Well, first of all, Elaine, thank you so much for the invitation. I'm so pleased to join you for this discussion and really enjoyed your opening remarks. The thing that I think sparked my interest in science was, first of all, my family. My father was a professor and someone who had written books at the age of 15. He was very sort of prolific and in our home, I had access to a microscope from the very earliest age. And so I have to say my home environment was one that was a lot about science. My mother was also in the biomedical field, so I have to say that I didn't stray too far from an area that my family certainly has been involved in, from academia all the way through to research. So that was really the spark. But I would say that experiencing certain situations in which family members or relatives and friends had become ill, I think while I had that original start in science, the idea of helping people to get better through discovery of mechanisms or approaches to treating disease, I think really also inspired me to pursue a career in biomedical research.
So it sounds like you started then kind of following in your father and mother's footsteps, going into getting your PhD. And there you did your then postdoc afterwards as well. So clear-cut academic. And back then, when you were in basic science, what was your area of research at those institutions?
Yes, absolutely. So I worked, first of all, for my PhD and first postdoc with Professor, now Sir Stephen Bloom in the UK at Imperial College. And the focus there was really around how the hypothalamus and the pituitary play such a critical role in so many of our organ systems and in our well-being. And so we were involved in isolating novel peptides from the pituitary and the hypothalamus that were involved in metabolic disease, in our response to stress and other physiological systems, or let's say homeostasis. Actually, that was a key focus for us. And that was fascinating. I think I sort of grew up at the time in my PhD / postdoc days where molecular biology was becoming this important approach, right, to thinking about how do I interrogate my pathway? Can I make different observations going beyond the pharmacology that we were doing in the lab. And so I actually made the decision to move on from a purely physiology pharmacology lab to the Ludwig Institute for Cancer Research, and a sort of huge right turn, if you like, to muscular dystrophy. The work I was doing at the Ludwig Institute was around deorphanizing a tyrosine kinase receptor to understand a driver of muscular dystrophy. It turns out that protein, NSK2, is also known as MuSK, is a key protein in muscular dystrophy. And it was actually during the course of that postdoc project that I realized Regeneron were going after the exact same project I was. And here was little old me, an individual at the Ludwig Institute, Cancer Institute in London at UCL, versus this team at Regeneron. And it was really at that point that it opened my eyes to the fact that there is this incredible work going on, not in academia, which is where my family assumed I was going to stay, but in pharmaceutical research.
So how did you, I'm beginning to get a sense of this then, at being at that interface between basic science and medicine, and how that started to shape your interests, and now I can see with this going on, you could have gone to Regeneron probably, you'd be close neighbor here for a long time. But how did you wind up at Fearing?
Great question. So I think my training to that point had been firmly in the area of endocrine physiology through my PhD and first postdoc training. But then when I moved to the Ludwig Institute, it was really about cloning out genes, ultimately coming up with fusion proteins. So combining this sort of endocrine physiology world with molecular biology. Ferring Pharmaceuticals, for those who aren't familiar with it, is a Danish company. It's, I think, almost 100 years old, if not already 100 years old. And the beginning was really around isolation of hormones and leveraging or harnessing those hormones to treat disease. They were at the time looking to figure out what are those next key hormones, key peptides that they could go after. And I actually had this interesting combination of endocrine physiology, but also forward looking into molecular biology and how we would run cell-based assays, for example, to test various new agents. I have to say, I went to the interview at Ferring thinking, that this was going to be a huge departure from my alternative plan, which was to get grants and run my own lab, unsure as to whether I would get that role. And actually, they told me I was unlikely to because I had no pharmaceutical history, no pharmaceutical background. And I think that is something that people are concerned about. If I haven't got any, sort of experience or background in pharmaceutical research, how am I going to kind of be accepted into that world? I actually returned to London thinking that this was not perhaps the path for me, and then ended up being recruited by Ferring. My first job there was on target discovery and also creating cell-based assays for a lot of new drug discovery projects.
So the big, one of the big chemotherapeutic agents that you're associated with in a big way is this drug called Firmagon.
Yes.
And I think it's used for prostate treatment. You haven't mentioned this so far. Did that start percolating? Because that's also a hormone-based treatment, right? Did that start percolating at Ferring, or was that then at Merck, or both?
Actually, that was work that was done at Ferring. So interestingly, gonadotropin hormone, which is obviously involved in the reproductive axes, although we were studying its potential for managing LH and other reproductive hormones, it became pretty clear that GnRH antagonists and agonists could play a very important role in reproductive cancers. We were attempting to create a stable GnRH antagonist that could be used in place of what had been traditionally GnRH agonists. So the cancer field has actually used GnRH antagonists for many decades to kind of dampen down the endocrine response, the hormonal response, and therefore shrink tumors. The problem was with the agonists was that in the process of treating people with an agonist, you would end up with an initial flare because you were essentially activating that pathway. It's actually driving further androgen response in this case. And so the patients had to go through this very difficult flare up in their disease before you had a downregulation of the GnRH receptor and ultimately a shutdown of that response. And so the hypothesis was, could you come up with a GnRH antagonist that still had great potency for the receptor, but that also was able to eliminate this flare? And that concept was proven, and our job was then to come up with a stable GnRH peptide that could be administered to patients. The unexpected thing that happened in that project was that the form of GnRH we came up with was so stable that it actually could be administered not daily, which would have been challenging for patients.
Yeah.
It could be administered on a less regular basis. So it became this exciting opportunity to have a depot of GnRH antagonist on board. And so that was a preclinical project. It ended up moving into clinical and ultimately became a successful drug. I just learned that actually that compound, degarelix, is being considered for a treatment for COVID-19, which I still need to look into that, but the drug is still being sold.
That's it. Now, this is amazing because I was going to ask you whether or not you could use your Firmagon in order to be able to treat other diseases, but I have to say I was not thinking about SARS-CoV-2. I was thinking about other types of cancers. So
Yes, well, indeed, other reproductive cancers have been studied using GnRH antagonists more broadly. There are several now of those GnRH antagonists that are being used for both male and female gynecological cancers.
Okay, okay. So this is exciting. I mean, I'm seeing you're starting the needle is starting to shift now into your mindset, but you're still clearly using and exploiting the basic science training that you've had to be able to go along these ways. But let me then jump to your next job, which is what, 2005, you moved to Merck.
Yes.
And tell us what inspired you to make that move. I'm sure Ferring would have been very happy with you if you had just stayed there.
That is true. By that time, I had moved to the US. I was working at the Ferring Research Institute in La Jolla, California. And my brief, as it were, when I moved to California was to essentially see if we could leverage the Human Genome Project to come up with new project ideas. And as part of that effort, one of the first questions we asked was, are there genes that we had not yet studied from a drug discovery perspective that were related to targets that were already under study for drug development? And at the time, a lot of news had broken about DP4 inhibitors. And we decided to search for homologs, if you like, of DP4, the enzyme that's involved in the breakdown of many peptides, in fact, and was a big project at Ferring. Through that work, we actually identified several novel DP4 homologs. And it was a sort of whole new class of these enzymes. It turned out, unknown to me, that Merck had invested very heavily in DP4 inhibitors for the treatment of diabetes. You sort of touched on something, that my whole world was based around basic science and preclinical science. But when Firmagon actually moved into the clinic, I had this kind of desire to understand what was happening to that molecule on the other side. And I actually, at the same time as we had identified this DP4 family of genes, had attended a training at Tufts University about clinical pharmacology, just like my move, if you like, from my first postdoc, where I said, okay, I need to learn molecular biology, I need to immerse myself. I felt the desire to understand clinical pharmacology. What is it that either makes a molecule extremely successful in the clinic or actually, leads to its demise? And it was at that event at Tufts that I met a lot of the faculty who were from Merck's clinical pharmacology group. So I went for an interview. They were looking for people with preclinical training to potentially join clinical pharmacology, which was a wonderful opportunity at the time. And someone heard that I was one of the team that had identified a series of DP4 inhibitor homologs and inhibitors. And I ended up interviewing with one of the high-paced teams at Merck who were working on what we now know was Januvia. Januvia, which is one of the top-selling drugs of all time, was a DP4 inhibitor that was highly differentiated versus anything else that people were working on. And so I had the fortunate opportunity to meet a lot of people on my interview day. And luckily for me, I got to join Merck in a clinical role, which was very exciting for me. I really wanted to follow drugs into the clinic, but here I was joining Merck to actually take molecules into patients for the first time.
So are the drugs that you were developing, did Ferring have a collaboration with Merck then, or how did that come about? Or did you start working on totally different DP4 inhibitors?
Actually, I joined Merck in Pennsylvania, working in neuroscience. So I did not end up following the DP4 project. I was fortunate that was kind of a sort of a way for me to get to know a lot of people even before I joined the company. But in the end, I ended up joining and working on areas that frankly were completely unfamiliar to me. I had not until that point worked in neuroscience. My first project
So really making that transition then to actually maybe not a transition, but really taking it from one area of drug discovery and development to the other area of drug discovery and development. And that must have been very, very exciting. So now I can say the same thing. Merck must have been delighted to have you there. And you worked there for quite some time. But then 2018, something about Schrödinger prompted you to consider switching yet again. What was that? What was that all about?
Yeah, it's interesting. So first, I guess the idea of embracing molecular biology took me from Imperial to the Ludwig. And then the Human Genome Project essentially led me to leave Ferring UK for Ferring US, to cross the Atlantic. My next transition, I guess, post-Merck, was the idea, and having now seen many, many projects in the preclinical space, I had the fortunate opportunity to lead a therapeutic area at Merck, where we were going from the seed idea all the way to supporting very late-stage programs. I had a role in business development and licensing at Merck in my last few years there, working with Roger Perlmutter. And so by that time, I'd seen a lot of discovery and early development projects. And the desire was, to kind of consider, can we speed this all up? Right? We're sort of, patients are waiting for us, and can we move faster? And in my role at business development at Merck, I was also responsible for looking at enabling technologies. And that was, what does the lab of the future look like? How can we move faster, all the way from biomarkers to some of the fundamental methods that we use? And I came across Schrödinger. Schrödinger was a very long-standing company in New York, 30-year company. And after many years, they had figured out how to code into software some of the very fundamental principles about how compounds bind to proteins and how to optimize those compounds. And what was astonishing to me was that they could do this not over the course of 3, 5, 10 years, which is how long it takes sometimes to develop a starting point for clinical study, but they were doing this in this alarming 2 to 3 years. And so I really wanted to know more. I spent a lot of time with the CEO, Ramy Farid, and we actually had a pilot project at Merck, which allowed me to see firsthand that this is real. And I was so excited about the potential not to apply this on just the one-off project, but to consider, can we build an entire pipeline that is based on using this technology, and can we show that we can really move this fast? And so that, coupled with the fact that I like building things and I love leading teams from the very sort of first moment of idea to a fully functioning organization, those two things appealed to me, and I ended up joining Schrödinger to help build the first Schrödinger fully owned internal pipeline.
How exciting. And also, I'm also thinking, as we're talking about this, that there are a number of different laboratories at Rockefeller who focus on cryo-EM or focus on small molecule screens. And now there's this third component of a machine learning aspect of drug discovery and development. And maybe you could give us a little bit of a context, since some of the students and postdocs are working in labs that are using these other approaches, and and crystallography approaches, of course, as well. Maybe tell us a little bit about how the AI machine learning fits in that scope.
Yes, absolutely happy to do that. I think, first of all, maybe just say a couple of things. Machine learning, otherwise known as even a regression plot or a structure activity relationship, those have been around for decades. And so I would say, first of all, that the things that Schrödinger are doing are really focused on first principles methods, physics-based methods. The issue, though, is that those physics-based methods and the computation required to understand not just one molecule and how it binds, but millions, if not billions of molecules and how they bind to that protein. You mentioned cryo-EM, X-ray crystallography. We take a structure of a protein and we basically look at the molecular dynamics, the molecular motion of that protein. So we're looking at a moving structure, but we're also looking at how, as that protein moves and as water molecules enter and leave various pockets on that protein, how do molecules therefore compete with those water molecules? How do they move in and out of the structure? That requires fundamental physics, but in order to calculate all of those things, you need enormous compute power. And the way that machine learning is integrated at Schrödinger is somewhat different to what I think people have been hearing about AI and machine learning, which is really about taking existing data and seeing what you can learn from that training set, seeing if the machine can identify signals in that training set that help you with drug discovery. We're doing it a little differently. We're using physics-based methods and we're using machine learning to really scale what we're able to do based on these computationally very expensive methods. The good news is that I sort of rebranded AI as augmented intelligence. It's about how much more can humans do, how much more can chemists do, powered by machine learning, in our case, by physics-based methods complemented by machine learning. And I think the truth is that our medicinal chemists at Schrödinger feel that they have this new superpower, which is understanding something that we can't see, which is these interactions that are at the atomistic level. It's funny that you mentioned physics in your introductory remarks. That's the story of Schrödinger. It's a physics story that we essentially layer machine learning on top of.
Oh, interesting. So I noticed in taking a look at the website for Schrödinger that there are a lot of it seems like a lot of collaborations with academics.
Correct.
And that's very interesting also. Certainly it's a characteristic of, say, Genentech or Amgen, and I think people haven't viewed it so much with regards to the systems biology and thinking about it here, but it's starting to, from what you said, it's starting to make sense. And I wonder if you could maybe mention to us a few, I don't know if you're at liberty to mention maybe something in the past of some very exciting collaborations that turned out to fruition.
Absolutely. So first of all, I'll say that Schrödinger was born out of an academic lab. So Rich Friesner, the founder, a famous theoretical physicist from Columbia, actually, really sort of tells you that the academic connection at Schrödinger is very, very strong. Over the years, it has remained that way. We're a science-driven company. We publish hundreds of papers, but we don't do that alone. We collaborate extensively with labs all over the world who are at the cutting edge in their fields, whether that's protein structure refinement, improving upon these physics-based methods. And so, yes, academic collaborations as well as collaborations with other pharma and biotech are key components of how we work. In terms of one of the more notable collaborations, certainly before my time, the idea that you could use computation to disrupt or advance our ability to make drugs, that was questioned. I think on the cover of Time magazine in the late 90s, the idea that computation and AI was going to disrupt drug discovery, the follow-up to that was a lot of disappointment. However, through a collaboration, actually it was a company that was sort of born out of Schrödinger. This is a company called Nimbus Therapeutics. That company was able to identify an allosteric binding site on a protein that the whole industry actually had been going after. And this started just a sort of collaboration between it was based on an academic paper actually, that had shown that there was this peptide that bound to an allosteric site on ACC, ACC being an enzyme involved in lipid metabolism. So from that paper stemmed a very important project, because the peptide that bound to that allosteric site allowed for a starting point for one of these physics-based chemical space enumerations. And that project was the first computationally designed molecule, not only to grow up in a biotech called Nimbus Therapeutics, but ultimately led to one of the biggest transactions in biotech history, which was the sale of that ACC inhibitor to Gilead. That molecule is now in phase 2 with the hope of treating NASH patients. And so that shows you that the sort of combination of academic research, the physics-based methods, the work Schrödinger was doing, and now this biotech that's based in Boston, that collaboration, I think, led to something that we hope will one day be approved to help patients.
So it really sounds, Karen, that you've got the best of both worlds now. You're literally able to interface with academics, able to interface with the pharmaceutical and the biotech, and able to do it all a lot faster.
Correct. With
Schrödinger. Sounds like the ideal job for you, and maybe for many of the postdocs and students in the audience today. And I wanted to, before I turn it over for questions, I just wanted to leave the thought to the students and postdocs and all of the rest of you who are in the audience of this. I think we're seeing this picture of someone who started out in a family with a lot of support and ended up becoming fearless as a consequence, perhaps. And that fearlessness and sense of, it seemed like you got the sense very early on that you could do whatever you wanted to do, and maybe to take that all the way through to keeping an open mind and the willingness to look at drug discovery through a different lens. And you've changed your lenses a lot during the course of your career, and I think that is a real inspiration for all of us in the audience today. And I think this might serve as a good time to open it up for questions. There's a question here from Andrea Morris, and she says, great conversation. What are your thoughts on industry postdocs? So this is probably coming from a graduate student.
Actually, I'm a huge fan of industry postdocs. As I mentioned, I ended up competing with Regeneron on a project. So I had no idea at the time, but the Merck postdoc program, for example, that was launched while I was there, and I had the pleasure of welcoming several postdocs into that program. I think for those individuals that got the opportunity to participate in that, and since then I've ended up hiring many people who've done these types of industry postdocs, what it does is, I think, to borrow your words, Elaine, it does give you a sort of an accelerated lens on what's involved in drug discovery, what's involved in various aspects of how a large or small company works. For many people who don't get that opportunity, they have to then either work in that field for some years or wait to be able to get that view. And so I'm a huge fan of them. I actually think that they're competitive for a reason, because I think it really does give you this accelerated view, and it gives you the opportunity to decide if you think that's something that you ultimately want to do. Nothing better than immersing yourself for a while to be able to make that decision. Is industry for me, or is it something that I would leave behind and go back to more of an academic career? So I'm a huge fan.
Great. Here's another question from Manda Hoford, asking, what do you think is the biggest barrier for applying Schrödinger methods to developing peptide therapeutics?
An excellent question. So I think you're probably aware that many peptides, physiologically, the cognate receptor for many peptides are GPCRs. GPCRs have formed the basis for some incredibly important drugs. The way in which those drugs were identified and the way I was involved in GnRH was very empirical. Keep testing lots and lots of peptides, synthesizing them one at a time. It's a slow and steady process. We need crystal structures to apply Schrödinger's technology. We need either a cryo-EM or an X-ray crystal structure. And both purifying and getting a structure of a membrane-bound protein is, as I'm sure you're aware, very challenging, but not impossible. In fact, we're excited at Schrödinger to see the number of ion channels, GPCRs, complex membrane-bound proteins that are now being crystallized or being solved by cryo-EM. So the challenge really in designing peptide therapeutics has traditionally for Schrödinger been around access to structure. Our software and technology works just as well on peptides, biologics, small molecules, macrocycles, but it's really about access to the crystal structure and an understanding of structure function that presents the challenge.
So Lulu Wang, one of our Board of Trustees members and one of the people who have really been behind the Women in Science WEF, asks for you, having seen how drug discovery can speed up dramatically, do you think this pace is just the beginning of a massive acceleration? And if so, what will be the different contributing factors in the future versus what has contributed in the last 10 years? Technology, scientific procedures, etc.
Yeah, super question. I think we are at the dawn of a true collaboration between biology and technology that has been ongoing for decades. If you think about what was possible with the Human Genome Project, so if we look back 10 years, actually, I think it's almost 30 years now since that project began, think about what we now know about the human genome that we didn't know when I was a postdoc. We now know an immense amount about the human genome, not just at the population level, but also now at the individual level. We are now getting single cell sequencing. I mean, these are all just incredible advances. But looking forward, I would say that truly integrating our ability to understand the patient, to understand the patient's disease journey, I think that's about to change, aided by technology. If you think about clinical trials and why we have such a high failure rate, one is related to the fact that we have challenges in developing the best possible molecule for the cognate target. That's something that we're clearly making headway on now with crystal structures, protein structures, and with technology like Schrödinger's. But think about the question, what about understanding who should and shouldn't be in a clinical trial, and are they likely to respond or not to that therapeutic intervention? We now have, I think, at our disposal, much better tools, and will have much better tools to understand a patient's pathophysiology from a multi-omics perspective. We're not quite there yet, but if you think about pathology, if you think about biomarkers, if you think about our ability to sort of interrogate the onset. Grail is a new company out of the West Coast that's now being, they're now testing the ability to measure cancer cell DNA, cell-free DNA. I mean, these approaches, I think, are going to have a huge impact on our ability not only to design better drugs for the right patient, but also to decide, are we at the right point in their disease process to have the maximum benefit? So I could talk for hours about this, but I think biology and technology coming together with other disciplines like physics, I think that we're at the cusp of this. It's super exciting.
Oh, nice. So Karen, this question comes from Marnie Imhoff, who is our Chief Development Officer at Rockefeller. And she says, I understand you have an interest in science education for young people. Can you tell us a little bit about your work in this area with your MyTech Learning, I assume?
Absolutely, happy to do so. So actually this stems back, and I'll just take a moment to tell you why I thought starting something like MyTech Learning was so important. When I was a PhD student, I was asked to give a lecture to students who were around 8 to 15 years old. This was in an inner city area of London. I was fortunate to live in the leafy green suburbs with my family, but I realized as I moved into London that obviously there are people who don't have access to the kind of education, opportunity, family background that I did. And so I started mentoring with the Prince of Wales Trust. And as part of that, I actually gave a lecture to this inner city school, a set of school kids. One of the children put her hand up and said to me, "You're a scientist?" She couldn't believe it, first of all, because I think she was familiar with the picture that you showed at the very beginning, that scientists have the elderly men with white spiky hair. And so she was confused by that. But she also said something that stuck with me. She said, "I don't think I could ever be a scientist." And I was stunned by that. She was 12 years old. And I asked her why. And she said, "Well, I don't know. And before I met you, I don't know anyone who's a scientist. And I don't think I'm smart enough." And I was floored by that. I was in my early 20s, and I swore that one day I was going to find a way to reach out to kids who are doubting their either interest in or ability in science. And that what we needed to do was to bring children closer to those exciting moments we have all experienced as scientists, those moments of breakthrough and discovery. And for me, that's always been about being in the lab. So I wanted to bring a program together that brought young children into the lab to run experiments. I think that's what it's all about. That's where a lot of people's passion comes from. And so that was the project that we started around 2010 with sort of small lectures in schools, little experiments. I ended up, between my time at Merck and Schrödinger, actually briefly opening a lab for kids to come in and experience what it's like to run experiments, and all the way from 5 years old up through mid-teens. And what I learned was that actually at school, unfortunately, a lot of children are not getting enough time running experiments. And so that's what that project was about. And again, it brought together these two themes of technology, biology, chemistry, physics. How do these things all interact?
So the aim is that 10 years from now, when the New York Times comes up with that front page article, they will say, when asked to draw a leader, they will draw a woman, typically.
Exactly.
So,
that's the hope. That's the hope. And maybe they won't be typecast as a biologist or a chemist or a physicist, but this is going to be someone who at least can understand the interdependencies of all these different amazing scientific fields.
Great. We have a question from an anonymous attendee. He or she says, you mentioned the important role of mentors in your work, especially when you undertook things that were new to you. Did the mentorship evolve, or did you seek it out? What would you recommend as someone just starting out as the best way to identify and connect with a mentor while pursuing new challenges?
Another great question. I always tell people that I'm mentoring, I take that very seriously myself, that you can't really expect necessarily leaders to find you. Sometimes they do. but I think it's really important to yourself identify what are those areas that I'm least sure about? What are those skills that I think I lack? And to proactively go out and find people who you think display those characteristics in ways that you would like to emulate. And so what I had done during my career was, as I discussed, I always like to be on the steep part of a learning curve. I always like to embrace technology, but I also love to focus on things that I'm not good at or that I need help with. And so seeking out individuals in companies, inside my own company, outside the company. It's so much easier these days with LinkedIn than it has ever been. But literally seeking out people who you think can help you with those areas for development. Mentors do play, I think, a huge role. I think it's not just about asking for help, but seeing how you can help them. At Merck, which is a almost 100,000 person company, I would often cross what would traditionally be boundaries to go and learn something or to go and help with an initiative that the company needed. And through that, I got exposed to a lot of things and a lot of people. And so I think, yes, I would recommend seeking out mentors who you think can help you grow.
Yeah. Great answer as well. So another question from Anne Spreel, and she asks, how do you recommend to handle, this is a good one, how do you recommend to handle the challenge or conflict between the academic need to publish with the private sector requirement to capture return on investment?
I understand the problem statement. I actually just ran with some colleagues from both academia, biotech, and pharma an NIH symposium on target validation. And I think one of the key themes that came out of that meeting was that the construct in academia where, and we've all been there, the idea that your papers are really a litmus test of your success, I think has over the years kind of driven a slightly different incentive system in academia to the incentive system in pharmaceutical industrial research, where there is this almost vicious focus on return of investment. I think neither one is perfect. The question is, how can we bring those two things together for the common goal of identifying validated, tractable pathway biology that will in some way benefit patients? And so what I would say is that I think there is challenge, there's conflict there. I think that's good because that can often lead to great things. But what we're finding is collaboration between industry and academia, where we bring these disparate, perhaps, mindsets together. And I think you mentioned it, Elaine, in your opening remarks, thinking about the industrial, well, let's not even say industrial, the applied endpoint, if you like, for a potential therapeutic idea. And let's at least try and speak the same language. What is it that in industry is considered a great target? What does that validation package look like? What would a pharma company, for example, prematurely give up on an idea that an academic group perhaps might see some promise in? Can we have that conversation in order to arrive at a middle ground that actually ends up being beneficial for patients? So I would say, yes, there's a lot of challenge and conflict, but there's also a huge amount of opportunity through collaboration.
Great. So there's another question from an anonymous attendee. Karen, your journey in science is so fascinating and your fearlessness is inspiring. What have you not done yet that you'd like to do?
That is a great question. I think one of the areas that has almost inspired my journey almost in, I would say, one thing I didn't say, my father became very ill when I was a postdoc. And there are certain areas of disease that get an incredible amount of attention. Oncology, rightly so. People are short on time, and it's absolutely critical that we work to find medicines to help them. But there are other areas of unmet medical need where there are patients suffering a different type of a different type of disease. One of those areas is kidney disease, which ultimately took my father away. And I have not yet worked on a project in kidney disease where I think we are making serious headway. So whether it's kidney disease or some other disease area where we are unfortunately unable to help people, there are several of those, but in particular kidney disease is one that sort of stays close to my heart, that I would like to be able to say, we found something that will work and that can help people. So yeah, the journey is never over as a drug developer or drug discoverer, but that's one that I personally would love to be able to accomplish at some point in my career.
Maybe related to that, you mentioned initially, I think, about Firmagon and possibly being used for COVID-19, but let me take you to your current Schrödinger position and ask you what kinds of things your AI approaches could have been doing along those lines to be able to come up with advances for therapeutics for COVID-19, for SARS-2-CoV.
Great question. So how could we work at a company that claims to accelerate drug discovery without jumping into this incredible space where we need something urgently? We actually, at the very beginning of this outbreak, had an enormous amount of inbound interest from both academia, biotech, and pharma to see how we could help. We actually decided to join ranks with two other companies, Takeda, Gilead, actually Novartis as well as Google Cloud, to see if we could accelerate the discovery, not of vaccines, but of therapeutics, antivirals. And the idea there was, can we cut down the development time, or discovery time, I should say, for a novel antiviral therapeutic? And so what we've done is Google donated, I think it was 16 million hours of computation time on the cloud for us to be able to run these enormous chemical space calculations to look for novel agents that work as antivirals. That project is still going on. It is a philanthropic effort between multiple companies. And we are leveraging this technology to see if we can speed up the discovery of these antivirals. Thankfully, vaccines will be the first wave of therapy, I think, or the first wave of prevention. But there will still be people who get sick with COVID-19, and we want to help discover those therapeutics. Yeah,
it's got to be exciting. It's got to be you're in the right place at the right time. And unfortunately, with the pandemic, it's not quite the challenge that you would like to see, or the world would like to see, but so important along those lines. Maybe the last question, since we are now running out of time, but might be that you often hear about a lot of challenges that women face. Certainly women have faced in academia over the years, and I'm sure you were experiencing that. There must be even more challenges for women in biotech pharma industry. And so I wonder, I'll turn it around rather than to ask you to describe all the things that you've experienced in your life growing up and with regards to this, but maybe turn it on a positive light to say, in your experience on both sides of the world, where do you find that women are particularly poised to become leaders and effective leaders in the pharma biotech industry and in academia as well?
Yes, it's a good question, and I do think that there is something to be said about an individual's desire to be successful. But I think what ultimately powers successful projects is the team effort. And I would say that, for the many teams that I've been on, women, I would argue, have a desire to bring the group with them, both in terms of explaining what the problem statement is, to recruiting the people, not physically recruiting in terms of HR, but finding the people who can help them and admitting that they need help. I think women in general are pretty good at that, recognizing where they need to partner to be successful and bringing the group with them. And that group effort, that group ability to bring all the ideas to the table, not just your ideas, but the ideas of everyone, to have the patience to listen to others, whether it was in an academic setting, a biotech setting, or even a pharma setting. I do think I've observed actually both men and women do this well. And those, I think, were some of the most successful teams I worked on. But I think many of the teams that I've seen led by women, it's their ability to listen, absorb all that information, and take the whole team with them, I would say, is a kind of power that we have.
On that note, this has just been such a wonderful conversation. I've so much enjoyed getting to know you, and as the audience has really enjoyed getting to know you and really being inspired by such an amazing career and the ability to not just try new things and venture into new avenues that have no blueprint, but also to really have the desire to make a difference, whether it's making a difference in terms of STEM with your company there, or whether it's making a difference with regards to human health and well-being. And I just think it's certainly been a wonderful start to our WEF conversations, and really want to thank you, Karen, for sharing your insights with us. And you've certainly given our audience a lot of good food for thought, and we're delighted that you could be our first speaker today.
Thank you very much. I really appreciate the invitation.
And I'd also, before we head off, I would also like to send our gratitude to the supporters of the Women in Science Entrepreneurship Fund again, who have really helped to launch this important series. And please, to the audience, please check the website for the Women in Science WEF in the coming days, and there'll be more information about the next webinar. And lastly, I'd like to thank our audience. There are still questions coming in, and unfortunately, we just had an hour to function at this on this venue today. So really want to thank the audience for very thoughtful questions. Be safe and have a wonderful evening.